All right. Hi, everyone. Thanks for joining us for our installment on development for the webinar series that we've had now for almost 2 full seasons. For those watching later and for those watching now, just a reminder that we will have this set of talks as well as previous talks up on our YouTube page. So if you are watching later, hello. If you are watching right now, if you have to leave early or you missed something, know that you can go revisit the material. I am very excited today to have an amazing colleague giving a talk. We have Attila Keresztes here, and he's going to be giving his talk, A Critical Look on the Development of Hippocampal Computations Supporting in Memory. I'm going to stop share so I can read his nice bio real quick. So Attila was first trained as an experimental psychologist at Eötvös Loránd University in Budapest, Hungary, and then he received his PhD in psychology and cognitive neuroscience from the Department of Cognitive Science at Budapest University of Technology and Economics in 2014. He spent his postdoctoral years at the Center for Lifespan Psychology at Max Planck in Berlin, where he worked in the cognitive and neural dynamics of memory across the Lifespan project. And after wrapping up his postdoc, Attila has started his own lab in Budapest, the Hippocampal Circuit and Code for Cognition Lab. And this is also currently a Max Planck Partner Group. His work aims to uncover the brain memory couplings across the lifespan. And really, I could not be more thrilled that he accepted our invitation today because he has such cool work that he has previously done and that I know he's sharing today that was recently officially accepted and published just over a month ago. So with that, I am going to hand it over to Attila, and after him, I will be giving a little bit of, I will be giving a talk. It's going to be more conversational. So Attila is going to be the heavy hitter with the science today, but all right, all you. Oh, still muted.
The necessary tech glitch. So
definitely.
Thanks for the kind introduction, Kelsey. You see only my slide and not my notes, right?
Yep, looks perfect.
Cool. Okay. So I really feel honored and actually very excited to present my work at this webinar series, which I have been really enjoying a lot and have also been learning so much from it. Before going to this, getting to the actual scientific content, let me say a few words about us. We are a relatively small and new lab with 3 PhD students and a couple of part-time research assistants. And I have to say, we have spent half of our lab lifetime under COVID lockdown, which probably has a vast effect on our lab life and identity as well. And as Kelsey mentioned, since 2021, we are a partner group of the Max Planck Institute for Human Development in Berlin, Germany. And specifically, I'm collaborating there with researchers, including my former colleagues and mentors within the Center for Lifespan Psychology. And the work I'm going to present is analysis of data that I collected there during my postdoc years. The research center where we are was created in 2013 in Budapest, as Kelsey said, in Hungary. And I started my lab at this site at the Arrow at the end of 2018. And just as a side note, Budapest has a continental weather with very hot summers and very cold winters. It's also almost 30 degrees. There was 30 degrees today. But luckily, the city has a vast number of hydrothermal caves, which we really like, that feed waters into the many baths we really like to visit. This is the Széchenyi bath I really like, and this is the Gellért. And despite my many visits, I needed a friend, a fellow HSGer, actually Drew Bender, to make me see that the Gellért, one of the most famous baths, has plenty of hippocampi on its walls with wrought iron. So, which brings me back to the content of the talk. So why take a critical look in the first place at anything? Well, I'm going to state the trivial. Falsifying parts of a theory can help us adjust that theory, and this is what leads to progress in science. And of course, this is trivial, as I said, but we really feel, I mean, we all feel, I think, that this is pretty hard to do at our own individual scientist level. And at least to me, I find it super inspiring and giving me chills when I see theory updates happening in front of my eyes and the people behind these updates. And one great example I really liked was presented here in this webinar series as well in April by Menno Witter. Dr. Witter showed a summary of their own results from the past few years, questioning a theory that had been supported by data from their own lab from the 2 previous decades, two decades earlier. So basically what he showed is that given novel data, that the perihippocampal cortex has extensive connections to both the lateral entorhinal and the medial entorhinal cortex. The earlier idea that the perirhinal cortex and the lateral entorhinal cortex form a neural pathway for coding object information, and the perihippocampal cortex, postrhinal in rodents, and the medial entorhinal cortex forms a neural pathway for coding spatial location information, may be just simply incorrect, and that it needs to be adjusted. And this is currently leading to novel theories that Dr. Witter said will be published in a new Hippocampus book edition, hopefully at the end of this year. So let's see a theory that I want to scrutinize. It's that pattern separation, hippocampal pattern separation, supports memory in humans. And this has to do with development of memory. So initially, computational models were informed by neuroanatomy and formulated the hypothesis that the hippocampus, and specifically the dentate gyrus, may be able to separate similar representations. Later, rodent studies, here's an example from the Logitech lab, showed that cells in the dentate gyrus compared to cells in the CA3 region showed large changes in their firing pattern, and relatively small changes occurred in their input. Later again, a few human neuroimaging studies with high resolution functional magnetic resonance imaging suggested that the human dentate gyrus in CA3 compared to other subfields may be highly sensitive to detect subtle changes in stimuli. And some further studies also linked variations in dentate gyrus CA3 activity to discrimination of highly similar memories. This was implied to have a role in segregating or separating memories that are highly similar. So for instance, the way the location of your car parked yesterday versus the location of your car parked today. You're always doing this smoothlessly, potentially aided by hippocampal pattern separation of highly similar inputs to your spatiotemporal memories. Spatiotemporal, the region that deals with spatiotemporal memories. And so together, the rodent and human evidence for the computational formulated idea of pattern separation led to the birth of this fascinating research program that I think is fascinating because it tries to bridge the gap between neuroscience and behavior in the memory domain. So how does pattern separation change during development? This is one key question in my research. One initial study by Zoe Ngo and colleagues suggested that relational memory but not mnemonic discrimination reaches adult levels by 6 years of age. To measure mnemonic discrimination, they adapted a task developed by Craig Stark and Brock Curven, the mnemonic similarity task. In this task, participants see a series of images of objects during an incidental encoding phase of an experiment. And then at recognition, they see identical images or just slightly changed images of things they've seen before during encoding, completely novel images. And they are asked to indicate if they have seen exactly the same image or kind of the same image, or they have not seen this image at all. And mnemonic discrimination is then quantified as the proportion of kind of same responses to slightly different pictures minus kind of same responses to completely novel pictures. Other evidence for ongoing development of pattern separation was provided by a study that showed spatial memory discrimination development between ages 2 and 4. And importantly, histological data from rhesus monkeys had suggested that the dentate gyrus is the latest piece integrated in the hippocampal circuit following a relatively late emergence and integration of the CA3 region in the hippocampal circuit. And initial evidence from human high-resolution neuroimaging from Getty's lab, sorry, there's a typo, in a paper by Lee and colleagues in neuroimaging, by Lee, Getty, and Ekstrom. This high-resolution study provided evidence that children may show a similar pattern of protracted hippocampal subfield development. In a study published in 2017, we showed that volumetric age differences in hippocampal subfields were indeed associated to mnemonic discrimination in 6 to 14-year-old children and young adults. We used highly reliable manual segmentations of high-resolution MRI images of the medial temporal lobe to measure volumes of regions within the medial temporal lobe. 4 of them, namely the dentate gyrus CA3, the subiculum, and the CA1-2, and the entorhinal cortex. And we found a positive linear age association for dentate gyrus CA3, for CA1, CA2, and for subiculum among the children sample. So this is from 6 to 14 years of age, and this is the young adult sample. And across the full sample, we found a linear age association for dentate gyrus CA3. So this suggested indeed that the most protracted changes may occur in dentate gyrus CA3. Importantly, using multivariate statistical tools, we showed that the heterogeneous pattern of hippocampal subfield development captured in one score and largely driven by dentate gyrus changes, dentate gyrus maturation, was related specifically to mnemonic discrimination measured in a similar task to the mnemonic similarity task I've shown you before, or in an adaptation basically of that task. And we showed that this relation between mnemonic discrimination and hippocampal maturity was specific to mnemonic discrimination, and it was not, this hippocampal maturity score was not related to other memories, performance on other memory tasks. So based on these mostly cross-sectional evidence, we have suggested that the uneven maturation of the hippocampal subfields is something that may drive an uneven maturation of pattern separation and completion. And this in turn may be behind the widely observed phenomena that early in life, children tend to extract commonalities of the world surrounding them rather than, or at the expense of, memorizing specifics of memories. To corroborate these suggestions, before all else, of course, we need longitudinal data. So caveat 1 is that there is scarce longitudinal data. And up until 2022, there have been only 3 studies looking at actual change in hippocampal subfields. So here, in a very not so nice table, these arrows indicate the age trends observed in longitudinal studies for different subfields with different ages and different extent of the hippocampus assessed. So these included different age ranges, different methods, and different results, importantly. Now, I have to note that cross-sectional data is also very noisy and looks a bit mixed. And so this, again, not so nice table, sums up cross-sectional age trends in the few published studies I have found by the summer of 2021, excluding studies that had largely overlap in their samples. And again, studies differed in age range, sample size, methods, and results. So just looking at regions comprising the dentate gyrus, you can also see that there's many different types of delineations, including the dentate gyrus. One somewhat consistent finding is that for these regions, significant age associations are consistently positive at younger ages. So even though there's these quadratic changes, initially there's an increase. And there's this odd one out study, but that study measured volume for only 3 slices, which makes up really very roughly 30% of the hippocampal body and really very roughly 10% of the total hippocampus. So the interpretation of volume as a measure of hippocampal, the measure of hippocampal volume in that study is different than in the rest of the studies. But again, this table nicely shows with these many colors and directions that the cross-sectional data is also just somewhat consistent. And here's an example from a study from Lars Nyberg's lab and colleagues that really nicely demonstrated that longitudinal and cross-sectional measures can disagree to the point of suggesting opposite signs of change. Here, longitudinal and cross-sectional age strands in frontal recruitment during task performance had opposite, positive in cross-sectional and negative in longitudinal age strands, so they had opposite signs. And to investigate the longitudinal and cross-sectional age associations between memory and hippocampal subfields in early middle childhood, I used data from a study that was involved that was led by Yee Lee Shing and Laura Raffington at the Max Planck Institute for Human Development. Of interest to this presentation, we measured hippocampal subfield volumes using high-resolution MRI in 2 waves when children were 6 to 8 years old and 8 to 10 years old. And this allowed us to assess cross-sectional and longitudinal age associations within a roughly equal time interval of 2 years. I will walk you through this part of the figure for the dentate gyrus CA3 to see this complicated, very colored figure and to understand what is represented on it. So the black lines show cross-sectional age strands for the 2 waves. The middle line, this middle grayish line, represents longitudinal change. Blue lines represent individual data points, with blue indicating increase in volume and orange indicating decrease in volume. And this C and L mean significant cross-sectional age strand and significant longitudinal age strand. So what you can see is that consistently with at least our own earlier findings and with some other cross-sectional data, we observed a positive linear age strand for the dentate gyrus CA3 and the CA1, CA2. It was only significant in the second wave, but the magnitude and the direction was very similar in both waves. But we did not observe any change in the dentate gyrus longitudinally. And moreover, we observed a decrease in volume in the CA1, 2, although this was not reaching the level of significance. In contrast, in the subiculum, we observed a linear increase longitudinally, but no age differences cross-sectionally. And again, consistently with observations in other studies, no cross-sectional age difference. Sometimes observed a little, we observed a little negative, non-significant age difference. But we here observed a significant increase in volume for the entorhinal cortex. So critically, we have found no evidence for increasing the dentate gyrus and CA3. And we did find evidence for positive change in the subiculum and the entorhinal cortex, which was kind of unexpected. And as you can see, there is basically no overlap in the findings across the cross-sectional. So there's no L here, there's no C here, and subregions differ widely. In addition, we measured memory task, spatial memory task, and associative memory task, and a mnemonic discrimination task. Again, the similar mnemonic discrimination task as shown earlier, the mnemonic similarity task, but here in a continuous recognition version of the task. And we saw that there was an improvement from wave 1 to wave 2, but there was no relation of this change to change in hippocampal subfield volumes, let alone to dentate gyrus CA3 volume change. However, to a surprise, we found that volumetric increase in subiculum was significantly associated with improvement on a spatial memory task. In fact, the limited available data on subfield volume memory associations is very mixed. And caveat 2, this null finding or this mixed findings could be due to the huge noise in our measures. First, behavioral measures of pattern separation and completion, including the widely used mnemonic similarity task, provide one outcome measure. But at the neural level, pattern separation and completion can both operate on any given input to the hippocampus. So they both contribute to every behavioral outcome. So as a result, single behavioral measures of each will provide highly noisy estimates. And you can add to this the additional noise in your measures that come with testing children. So structure function couplings may vary also across individuals and over time. So individual differences in structural measures may capture also a diverse range of functionally relevant individual differences. This is really nicely shown in Kelsey's study published in Cerebral Cortex in 2019. there, in a sample of 4 to 8-year-old kids, they showed that the association between volume of dentate gyrus CA2 to 4 region and mnemonic discrimination swapped signs at around age 6. Oops. So are adjustments in order to any theory and methods? I don't want to rush to conclusions based on the limited number of longitudinal studies, but the available data may suggest that we need to adjust our notion of hippocampal subfield development. And it may be that the largest functional changes do occur just below the age of 6. In fact, this should not come as a surprise given the animal evidence, which suggests that age 6, 7 years of age may be the latest asymptotes of neural maturational processes in the hippocampus. The data on change in subiculum volume and its coupling with memory development is consistent with findings of Kelsey's study. I hope she will present in the next talk. And this will bag and already bags novel questions about its role in memory development. The studies presented also identified methodological challenges. One solution to the issue of noisy behavioral measures may be the use of a latent variable approach, where multiple behavioral indicators are used to assess a latent construct. An obvious solution to the noisy structural measures, of course, is to add functional measures, but this is a hard challenge, and I think only a few labs will try to address. And for the moment, myself, I will also just focus on longitudinal measures of structure below the age 6. In addition to collecting data, given that the investigated changes are very, very likely to be nonlinear, we are probably better off with more than 2 measurement occasions if we go to the longitudinal route. So here I just flash a study in its pilot phase that is headed by Kish, my PhD student, that tries to address these challenges. This 3-wave longitudinal study will assess hippocampal subfield volumes and memory below the age of 6. And importantly, we will assess pattern separation and completion as a latent construct, or as latent constructs from the behavioral readout of 3 to 5 tasks, which tap into these neural processes. Here, pattern separation piece, I was having trouble finding my mouse. And we collect data for a bunch of studies and try to see for the moment whether we can actually, using some kind of structural equation modeling and confirmatory factor analysis, to see whether these can be grouped together to form latent constructs of pattern completion and pattern separation. So to conclude, I would like to highlight the large uncertainty in our measures of hippocampal subfield development and the concurrent development of hippocampal computations. And one thing that with all these open science and new initiatives for better science comes to mind is, of course, that we need to reduce the uncertainty as much as we can. And the methods harmonization efforts of the Hippocampal Subfields group is an excellent route to achieve this objective. Another important route that I believe Kelsey will also talk about is running studies in consortium. But yet another objective is to acknowledge uncertainty. And I would like to conclude the talk by advocating a provocative opinion paper that appeared last week in the journal Nature, co-authored by a colleague here at Eötvös Loránd University. And these authors propose a shift in our approach to analysis and publication, the multi-team analysis. They actually suggest that before a publication, we should invite independent researchers to analyze our data. We only give them our raw data and the research questions, and we invite them to be co-authors on a study that will show multiple answers to the same question using the same data. And this way, the published study will already incorporate estimates of uncertainty. And so I really believe that given the huge uncertainty of human neuroimaging estimates, especially of hippocampal subfield development, and the additional uncertainty of behavioral estimates of hippocampally supported memory development, I think that this may be this multi-team or many analysts approach may be a viable route to chart the potential developmental brain memory couplings that we're looking at. So with that, I want to thank you, and I'm looking forward to your questions.
Thank you, Attila. I love the way that you structured that talk. I've seen it, a similar version of it before. We were talking yesterday, very early in the morning before, so it was nice to see the updated version while actually awake. I don't necessarily have a question. I just want to echo your sentiments of, I think it's great to acknowledge the limitations of our data. I mean, we're both developmentalists, but it's not specific to development. But I really do think that by acknowledging, you know, the mixed findings that we have and the different approaches we take, we can move towards a better understanding of how and why our results might diverge. Because, right, the end goal is that we really can understand what the developmental trajectories of subfields that are important for cognition are. So I just get geeked out when I get to talk to other developmental people. But I will say, if anyone wants to chat in the chat box, we're going to have time. We have time for questions or discussions right now. I would offer people just to unmute and talk for a few minutes, but I also get nervous because I'm very much a control freak when it comes to those sorts of things.
I have a question.
Yes, go for it.
I just wondering, I mean, first of all, fascinating talk. And again, I agree with Kelsey, it's very well structured. And I really quite agree with your concluding point. The Wagenmaker's paper is very important. And this is, you know, true for all science, especially in the field of Alzheimer's disease. With Alzheimer's Disease Neuroimaging Initiative, we need to be doing more of this sort of team analysis rather than these isolated silos. But okay, so my question to you is, the hippocampal subfield maturation is fascinating, but of course this is occurring in parallel with the neocortical development. And I mean, do you have room in your theory for thinking about these things as parallel integrated evolution, things that are evolving or developing at the same time, I should say?
Yeah, definitely. And thanks for the question. I wonder, Kelsey, how I could know who's asking the question. I don't see
that was Taylor Schmitz.
Okay, thank you, Taylor. Thanks. So yeah, there is room for this in the theory, but not specifically my theory. It's just theorizing, any theorizing about memory development. So, for instance, many are studying how frontal maturation can affect hippocampally based memory development, especially the development of the interaction between frontal cortices and hippocampal or mediotemporal lobe regions. One specific instance of this is how strategic retrieval may develop, or how strategic encoding may develop as a function of changes in those connections between frontal and hippocampal regions. One specific question that we may be interested to look at is how specific hippocampal computations, so for instance, pattern separation and completion, may be biased by frontal expectations or predictions. For the moment, I have to say we have conducted one such study with all young adults, or older children, young adults, and this was inconclusive. So for my side, I would just, for the moment, I don't plan any further studies with children that looks at this, but there's papers from the Ulman Lab, I think, that looked at this. There's papers from Simaghetti or Yee Lee Shing as well that looked at development of these interactions and how they affect memory.
Could I ask one quick follow-up? Sorry. So yes, I agree. And do you think that sleep research, so I mean, of course, sleep changes across the maturational stages, and of course, sleep is heavily, regulated by hippocampal cortical interactions. And could you use that maybe as a biomarker of some of these things that you're talking about computationally during a waking state?
Yes, that's an excellent idea. And my former supervisor at the MPIB is actually conducting studies with one of her PhD students. I'm blocking on the family name of Anka Jöchner, and Markus Werkle-Bergner. They are conducting such studies with 4, maybe 5 to 7-year-old kids. So I'm really looking forward to their results.
And Tracy, I see you with your hand raised nicely. Go for it.
Oh, that was such a great talk. Thank you so much. And I love to hear people say, you know, 3 to 6 years of age is where the action's at. I totally agree. So it was just delightful. I had a question about if you could just speculate a little bit about what we might be missing with volumetric measures and anything that you have thought about or seen that we could challenge ourselves to do with younger children, either in terms of functional scans or diffusion scans that might tell us a little bit more about, you know, what's developing in addition to size.
Yeah, thanks, Tracy. That's a big question. So I actually already forgot the first part of the question. So basically, if I remember correctly, the first part was what else could develop, or what structural, what we may be missing with structural measures. Well, you also describe in your papers that we may not, I mean, with this shift, for instance, in the sign of associations between behavior and brain volume, we may just not know whether synaptic growth or dendritic arborization is ongoing, or cell neurogenesis is ongoing at a certain age, and to what extent these are ongoing, which would lead to volumetric increases and potentially improvements in memory. And at what point should we look at volumetric changes as signs of pruning or consolidation of a network, especially the hippocampal network, or perhaps consolidation of hippocampal frontal connections? So we should probably map and have theories about at different age ranges, what are those connections between brain volume and function. And I'm super excited by technological developments in the field. I know, for instance, that the Prisma scanners in Siemens have now inbuilt high resolution DVI sequences, but I also know that they are super sensitive to motion. So I'm really looking forward to experimenting with those sequences a little bit more in the future to see whether we can get at higher resolution of connectivity, potentially between longitudinal axis portions of the hippocampus or just other regions in the cortex and hippocampal subfields. So yeah, there's so many questions to answer. I think that the best questions are yet not answerable with the techniques we have. So we also need to, in addition to these, I think we really need to have crosstalk with rodent studies that look at infant development of hippocampal circuit and potentially have memory models in rodents that can answer these questions and inform further research in humans. I'm not sure whether that answered anything about your question, but I hope that as a speculation, this may be something.
Definitely. Thank you so much.
All right, I'm going to swap over, but we will definitely have time for conversation because mine is going to be a little bit of science and a bit of conversation.
All right, so our next presenter is Kelsey. You've been hearing from her all morning and for the last 2 years. My name is Valerie Carr. I'm one of the steering committee members of the HSG. And normally Kelsey's co-host for this webinar series would introduce her, but he is traveling at the moment. So I will do Kelsey's bio. So Dr. Kelsey Canada is a postdoc at Wayne State University in Detroit and the Institute of Gerontology and a member of the Lifespan Cognitive Neuroscience Group. She works with Drs. Noah Ofen and Anna Doherty, Side note, Anna just got married, so congratulations to Anna. Dr. Canada completed her doctoral training in cognitive and neural systems in the Department of Psychology at University of Maryland, College Park with Dr. Tracy Riggins, who just asked a question. And her research is focused on the development of the hippocampus and accompanying changes in cognitive abilities. So very much in line with some of what we just heard from Attila. She's most interested in episodic memory and the structural development of hippocampal subfields across early development. And she's currently working on characterizing how children's brain and cognitive development is modified by resources in their home and neighborhood environments. So today she's going to be telling us about the importance of longitudinal data and diverse samples in understanding hippocampal development. So take it away, Kelsey.
Thanks so much, Valerie, for taking Robon's position for the day, as he's on a train, and we were not going to risk Wi-Fi on a train if anyone has ever done that. So thank you, and thank you everyone for sticking around for hopefully good chat that we can have. That this will tie in nicely with what Attila just presented. Like I said, I've got a little bit of science today. Of actual data, but more of this is going to be broader stroke conversation of the way I've been thinking, and hope to talk with other people who are doing not only developmental work, but work across the lifespan, because some of the ideas I hoped would be useful. So it's no surprise to anyone here that the hippocampus is a heterogeneous structure. And it has functionally distinct subunits with the dentate gyrus, the chronomonas, and the subiculum, and that these are distributed along the long axis of the hippocampus into the head, body, and tail. What recently I've been spending more of my time on is considering how volumetric variations in these functionally distinct subfields are implicated in neurodevelopmental disorders, such as autism, ADHD, and schizophrenia. And given the fact that we have origins in development for disorders and links to hippocampal structure, it's also important to consider what the development of the hippocampus is. And so we know from work in non-human primates and in postmortem human studies that we have protracted development of this structure, where we think we've got the earliest maturation subiculum, followed by CA1, and then dentate gyrus with the accompanying recurrent connections to CA3. Sorry, like I said, light on science, but this next slide is largely going to be the version of what Attila just showed us with the table, is that we have cross-sectional work in vivo in children that suggests that there are volumetric differences in hippocampal subfields, but results are not fully consistent. So it's very difficult when you're trying to review the literature and get an understanding of what development of this critical structure is. If you're only looking at cross-sectional studies and trying to map them on one-to-one is a very difficult venture. And importantly, we also have longitudinal work that suggests that there's distinct developmental trajectories of subfields. I don't actually have a lot of this work here today, but it is largely, at least in my previous work, looking at development before the age of 6, where you see increases in these subfields that are robust. But longitudinal work also highlights the fact that there are potential disparities between our cross-sectional and longitudinal studies, which raises the fact that we do need longitudinal research to help better answer our questions, not only of what development of hippocampal subfields looks like, but trickle-down implications such as neurodevelopmental disorders and cognition. Okay, I'm going to switch a little bit to talking about environmental aspects in a study that I'm working on right now, and then I'm going to get into sort of broader picture of why I think longitudinal and diverse samples are really essential, especially in developmental research. So we know that the hippocampus is impacted by the environment. Importantly, the hippocampus exists inside children's or people's heads, and children don't exist in little bubbles. They have environments, they have social lives, and we know that their socioeconomic, so the resources that are available to them, has lifelong predictions, both on lower cognitive abilities and on a greater incidence of neurodevelopmental disorders, both of which are linked in part to the hippocampus. So this link between early life disparities, we can say, in children's experiences and outcomes such as neurodevelopmental disorders and lower cognitive abilities can partially be explained by the hippocampus. However, we don't know too much about socioeconomic status in hippocampus beyond the total hippocampal volume. So we know that variations in total hippocampal volume have been linked to household and neighborhood socioeconomic status. I'm not going to go into both of those too much today, but really relatively little is known about the vulnerability of specific subfields that are distinct in cytoarchitecture and function to socioeconomic disparities. And further, work in adults suggests that there are potential vulnerabilities that are specific to certain subfields for neuro and psychological pathologies, specifically in the dentate gyrus and CA1, more so than in the subiculum. And so I'm going to show you now a little bit of data, an initial foray into looking at socioeconomic status related to hippocampal subfields, and then move into why I don't think that's a good enough place to stop with what I'm working on. So in my initial cross-sectional foray, in 157 5 to 25-year-olds, we manually demarcated the CA1 through 2, dentate, CA3, and subiculum in the hippocampal body, and examined socioeconomic status in this group of children from the Detroit metro area using a composite measure of maternal education, paternal education, and incomes to need ratio. And so the goal of this study is just an initial concept check of, is there specific vulnerabilities of structure of hippocampus in a developmental sample to differences in socioeconomic status? And can I carry that out then to look in a larger, more representative sample, hopefully down the line? So just as a quick check, we see in this sample, if you look here on the Y, we've got our volume and here's age. In this sample of 5 to 25-year-olds, there is age-related variation related in the subfield where you have larger age, greater age related to larger volumes in the dentate gyrus and CA3, but you do not see a robust effect in CA1 nor subiculum. So this is in line with the table that Attila showed, where there is variation across, but it seems like there is protracted development, at least of the dentate gyrus, CA3, that seems relatively consistent across studies. But more importantly, in this initial foray, is that there seems to be unique susceptibility of CA1, 2, and dentate CA3 to socioeconomic status. So now we have volume again on the Y, but now we have this composite measure of socioeconomic status here on the X. And you see that there's a relation between the dentate gyrus and CA1 to variations in socioeconomic status, but not in subiculum, which again, subiculum we think is relatively early developing compared to 2 and would be less susceptible given the distribution of different stress receptors. So just some current thoughts. This initial foray is that hippocampal subfields are likely uniquely sensitive to socioeconomic disparities. And this trickle-down effect is that you likely have impacts on specific cognitive functions, especially episodic memory, which we know is also developing, and Attila just showed some nice work during the early development, but is also protracted as well. And I think that during COVID and having to challenge myself of how to think about my research and data and what's important when you can't collect new data, it really makes you question like what you want to look at and what you can think about, is that we have to consider the individual differences factors that we have on hippocampal subfield development. Because if we can look, in addition to characterizing typical development, we can try to look at avenues for targeted intervention to promote healthy cognitive and brain development. But we can't really do that in single cross-sectional samples. And so this is where I'm going to pivot and talk about, I think that in order to have not only a better understanding of developmental trajectories and the impact of socioeconomic status, but also that coupling of cognitive and brain development that Attila described, we need to have not only longitudinal samples, but representative samples. And all of us here are probably intimately aware that when we're measuring subfields in vivo, we have a lot of difficulty in actually measuring the structure in a way that is both structurally and functionally exact. And our measures are educated approximations. they're good approximations, but they're not going to be necessarily, if we had the histology, what we would exactly hope and want when we're measuring them. And we also have variations in the atlases used to define hippocampal volumes. This relates right to the harmonization effort of the groups in order to be able to better compare our findings. But specifically in developmental work, we also have this, not issue, but we also have this factor of the fact that we oftentimes examine things at different scales and at different age ranges, which can make it difficult to tease apart how development is occurring, when things are impacting development, and so on, especially when everything is lumped together in a cross-sectional research. So overall, I think that we're sort of limited right now within single studies, and it's hard to actually test the hypothesis of non-distinct, nonlinear trajectories that we think are occurring. This is mirroring what Attila said, and it's difficult to test sort of what would modify these trajectories. And so something that I've spent a lot of time thinking about over the last couple of years, and I hope that other people might get excited about too, is the idea of an integrated data analysis framework in studying hippocampal subfields. And the concept, which is not new, if you look, 2009 is where this sort of hit the scene more, mostly in addiction research, is that you can pool raw data from multiple independent studies. So this would be me and Attila being like, hey, let's have our data shake hands in order to have one data set. And then you're actually directly modeling your questions on this new pooled data set. And what this allows you to do is to examine research questions from multiple samples, so different samples, different collections within the same analysis. And critically, you can test new questions with this framework, hypothetically, this is my hope, that are not possible within individual studies alone, right? We're limited by our resources. We're able to test amazing questions independently, but we can't answer all questions with our limited samples. And so when you are in IDA, the idea is that you can pool together these samples to not only increase your statistical power, but you're going to increase your sample representativeness and then also the generalizability of the results that we have, which especially in development, we'd like for our findings to generalize broadly beyond just the small samples of children that we're able to bring into the laboratory. And so this is a little bit of stats nerding, but I think that you'll be surprised if you haven't heard of IDA, what the requirements are and how this might be very feasible, is that you just need to have your raw data from different sources that might be different measurements having measured the same construct. So it may not be an identical measurement between studies. This is going towards the fact that we oftentimes use different atlases, but we're all trying to use our different atlases to measure the same thing. That is hippocampal subfield structure. So we don't need to have identical measurements that are mandatory, but we do have to have this shared subset of measures that have theoretical and operational definitions. So I'm not going to try to use the definition of entorhinal cortex in one study as representing CA1 in another study. There's not that ability of mismatch. We have to have things at the construct level that are matching, even if the exact measurement is not identical. And so the way that I'm going to be trying to apply this to my work in the next couple of years is that by looking over development, we can try to test not only what developmental trajectories are of hippocampal subfields, but that we can also look at the modification in larger samples. So I like this image, but what I want to show you is that my plan in using this, and I hope other people maybe try to adopt this, and I haven't done this yet. So this is where it's sort of a conversational aspect, is that NICHD has signed off on funding me for the next 3 years. And I was lucky enough to have amazing collaborators. So Simona Getty in Davis, Tracy Riggins in College Park, Ali Preston in Texas and Austin, and then both Noah Offen and Anna Doherty in Detroit, willing to combine our data to try this integrated data analysis. So instead of having our independent study samples of 140, 165, 1172, and 201, instead of using IDA, what we would have instead is 678 4 to 25-year-olds with almost 1,000 scans collected. So we have this longitudinal data. And further, by pooling data, we can move beyond the representativeness in just a single area of the country and instead have something that would be much more generalizable, is my hope, to the rest of the population. So if you look here, right, we're at different parts. It's nice. We have a spread across the map. We have males and females. We have people who are of different racial and ethnic groups. And critically, for my work, is you've got a lot of variation in socioeconomic status across these sites as well. And so I don't have data for you for this right now, but I can share sort of the concept here. Is this is done in a latent modeling framework. And so you have integrated raw data in which you have the same ideas, right, measures. You've got something DGCA3, something CA1, and something subiculum that is shared amongst these studies. And you use a latent modeling framework to look at what is shared amongst these measures that give you measurements that you can then test the developmental trajectories across this 4 to 25-year-old age range, where you have, I don't know if I go back, I'm gonna not find it, you have overlapping samples that entire time. So there's longitudinal data covering that entire span in order to look at what trajectories are, and additionally for me, how children's environments are going to potentially modify those trajectories. So I think that this is an approach that I'm hopeful will work, because this would allow us to have more accurate estimates, right? Instead of having our pigeonholed studies, which are great, but also limited in their own right, we can try to characterize trajectories. And this would facilitate, again, this idea of being able to better say, this is what development looks like, and then identify deviations from that typical development in facilitating intervention of neurodevelopmental disorders or learning disorders, what have you. And critically, and what I'm hoping is exciting for the group, is that if this is successful, this would demonstrate a means to leverage our existing data together to answer critical questions that are not feasible in individual studies. So for me, I also was like, okay, people are going to ask me, like, why not something else that's maybe less time intensive and tedious and trying to figure out what goes and what doesn't go. So a really useful and increasingly popular in the time of COVID, where we couldn't collect new data, approach is meta-analysis, which are highly valuable. I here threw up a citation that was led by my colleague Morgan Botdorf that was in total hippocampus and episodic memory. So I'm not by any means disparaging meta-analysis, but they are limited in using summary statistics. We don't have the actual raw data in these analyses. So what we can test are not necessarily new empirical questions. We're going to be limited by what we can get from people who are willing to share their data and the summary statistics from that data. And for everyone here who collects any sort of data, you know that it's very timely and expensive to collect new data sets. And so the application of an integrated data analysis would leverage our existing high quality data, because all of us painstakingly try to get the best quality data possible, with an opportunity to make data more accessible and affordable, right? Collaborative science, I think, is something that at least I'm very promotive of. And this also, finally, would provide a potential complementary approach to harmonizing data. So I, as you know, part of the HSG, fully support and advocate for a harmonized protocol in order to better draw comparisons. But sometimes the atlas that we use is titrated to our specific research question and what is most appropriate to our samples. And so if it is a point in which we are still, you know, in part using, even within the harmonized, different definitions, being able to see what's shared amongst them and an integrated data analysis approach, I think would really be helpful, not only for developmentalists, but generally in order to better understand development of the hippocampus, of memory in general, overall, to make it more generalizable. So I really hope that others are on board. I know most people are advocate for longitudinal data, but I also think it's important to have representative data. And one way we can do that is by trying to integrate our data together. So with that, I just want to say thanks for your attention. We're going to have a few extra minutes, and I will stop my share so that we can all have a chat.
All right, thank you, Kelsey. I love that both of you finished your talks with suggestions for how to move the field further with new methodological approaches. So that was fun that you had that similarity. As Kelsey said, we still have a few minutes for questions. Like last time, you're welcome to put your questions in the chat, or you could just unmute yourself and ask directly.
I already asked Attila yesterday. I'm like, can we share data? And he's like, let's talk.
Oh, hi.
Hi, Noah.
Hi. So I'll just say, Attila, I missed most of you, but I'll catch it later. But the end was fantastic. And then Kelsey, great job in kind of getting a discussion up, not just kind of the data. And I think I don't have a question. But I was just not sure if everybody heard the general, like to reinforce a general theme, it would be really good if we can all share data and expand that effort that Kelsey's we're starting here in the US. It kind of made sense more broadly. There's absolutely no reason not to, and this is just the perfect audience to just make it very explicit that this is meant to be as inclusive as possible. For anybody that has such data as right now, Kelsey might be the right person to lead this, and it's almost like our test case to show how awesome, it sounds great, and we're very optimistic that it would look great once we have it, but then it shouldn't stay there, it should really be expanding. Great job. And Valerie, you're managing it very well as well.
Hi, Tracy.
I was trying to give other people a chance, so feel free to kick me off, anyone.
No,
I waited as long as I could. That was great.
Rosanna's not here today, so you can be our Rosanna for the day, where she's like, do I, don't I?
Yeah, that was great. And I think, of course, I'm a fan for pooling data. So, but I think we also perhaps should be mindful if and when we are starting new studies and new data collection, to sort of think in our mind, we might collect the data to answer a new question or for some new idea in the lab, but then know this is a possibility down the line, and perhaps be cognizant of what other information we're collecting at the time, when new data collection occurs. And I was trying to search for it really quick. I think in the journal Developmental Science, they had a suggested sort of demographics form that people start using, just so that we're all asking kind of the same questions, particularly in regard to socioeconomic status. Are you asking about maternal education, paternal education? Are you asking about income? How refined are your income brackets? Is that adjusted for the neighborhood or the city, the state, the country that they live in? So this is just to take your idea and run with it, and perhaps encourage people to think about that when they're even collecting new data.
Yeah, thank you. I agree. I'll have to, right now I have to make a please list for Noah's new renewal, R01, when she has people come back. Can we please get this? I think you're muted, Noah.
I was going to say some of it is kind of set in NIH has some growing efforts for harmonization of data, for data archiving, but just collecting of basic demographics has been, I think, standard is probably, 10, 15 years ago. So at least that is something that people have been doing more consistently because it was asked by the funders. But absolutely, there's more room to expand and kind of generate those harmonizations further.
Awesome. Well, I am not going to end it yet, but I'm going to stop the recording a second. So with that, I'm just going to thank you again, Attila, for your amazing talk. I really appreciate you coming and sharing that work and being open to having this conversation with me and with everyone else about how we can not only improve our own science, but, promote collaborative science and better science overall. That's the goal. We have one more session this season 2, which will be our trainee session. So keep an eye out. We're going to have 2 great trainee talks, at least on there. The third is a maybe, but that'll be great. As always, I'm always impressed by everyone who comes here, and I thank you all for your time and attention. So with that, I'm going to stop my